We're really interested in the use of a molecule that we denote called RNA that is similar chemically to DNA, the materials that make up our genes, but it's a little bit different, and historically has been a little bit more challenging in terms of turning that or targeting that as a anti-cancer drug, for example. And so we think nanotechnology can be used to deliver RNA molecules into cancer cells, and that's roughly what we're studying. This is a particular kind of research grant from NIH, and it's called an R15 award, and it's really to stimulate research for undergraduates, biomedical research for undergraduates. So one of the goals we always have in mind is giving a solid research experience to undergraduate students here in the Cell and Molecular Biology program. And with my co-principal investigators on the grant, that extend Dr. Wanakea in the chemistry department, Dr. Gosh in the physics, astronomy and materials science department, and Dr. Garrett in my department, biomedical sciences, that would extend to students in those different departments. So our research is multidisciplinary. It involves two biomedical scientists, a chemist, and a physicist. Our goal, and this is the overarching theme in our entire research program, is the binding and the stabilization and the delivery of therapeutic RNA molecules by nanoparticles. For an RNA to get into a cell, it needs a carrier. It needs to bind a nanoparticle. If the ideal thing is that that nanoparticle then shepherds the RNA into the cell. But it must protect the RNA from all the things within the cell that would seek to degrade it. So there comes the stability factor. And then the delivery, it has to, some of the RNA molecules that we study actually have to act in the nucleus, so the nanoparticle has an even tougher job. It doesn't just have to take things into the cytoplasm, the inside of the cell. It has to go even further and into the nucleus in order for that RNA to act. So there's the delivery component too. So this new NanoSite instrument is essentially kind of a microscope that is configured that it will allow us to essentially image nanoparticles. But what this does that is really unusual is it will allow us to measure whether a nanoparticle is bound by RNA molecules or protein molecules. Okay? And it has the great feature when we start treating cells with nanoparticles or nanoparticles bound to RNA nanoconjugates, as it were. It has the unique feature of being able to quantify the dose, particles per mil. That is huge for us because now we're able to do our science with much more mathematical precision. When we treat cells, when John, my graduate student, for example, treats cells with a class of RNA molecule called splice-switching oligos that we study, he now will do so much more with much more precision, with much more control. He knows how many cells he has plated down, and he knows how many particles those cells have been exposed to. Even better than that, he actually, using this instrument, will be able to measure those particles that have RNA bound to them versus those that do not. So we will basically be able to measure an effective dose as well. So right now, conventional chemotherapy is a very non-specific treatment. It works, does kill cancer cells, but it's also very toxic to normal cells. People get really sick when being treated by chemotherapy. So actually, there's a couple of benefits. One is, and actually these clinical trials are already going on all over the world, is that you could deliver conventional chemotherapeutics highly bound to nanoparticles. So you are increasing your effective dosage, basically. there's a lot of interest in marrying targeting technology with nanoparticles, because not only could you highly load the nanoparticle with even a conventional chemotherapeutic drug, but you could then target that specifically to the cancer.